Hi class, we're going to go over a few epithelial slides. These slides were taken from our lab. One of my former students took pictures of them like you guys have been doing them through the microscopes with his iPhone, and he sent them to me. So we're just going to go through them and just as a little review and see how you do. So let's just start. Here's our first slide. Now remember the characteristics of epithelial tissue, CRAP. So if you remember your CRAP, the C stands for cellularity. There are lots of cells. In epithelial tissue, there is no space in between the cells. The cells are just right next to each other. So lots of cells, cellularity. R, rapid regeneration. Rapid regeneration, they divide rapidly. A, avascular. Avascular means there is no blood flow through epithelial tissue. Polarity, just like the North and South Pole, there's a top side and a bottom side to epithelial tissue. The top side, or the apical side, of epithelial tissue always faces a free space. Nothing up here. There is nothing up here. This is a free space. This is called the lumen, free space. And then we have the bottom layer, the basal layer. The basal layer is connected to a basement membrane that is connected to the connective tissue below it. And remember, it is the connective tissue below the epithelial tissue that is very vascular, and that's where the epithelial is able to get its nutrients and oxygen by diffusion from the connective tissue below it. So that's just a little review. CRAP, remember your CRAP, and let's get started on our cells. So remember, with epithelial tissue, the first thing you look at, is it single layer? Is it only one layer thick, or is it two or more layers thick? If it's single layer, it's called simple. If it's more than one layer thick, it's stratified. Now on this one, here's the basal layer, here's the apical layer, lots of cells in between. Let's go a little bit higher. So this obviously is stratified. Lots of layers here. And the top layer here, if you can go a little bit higher, you can see the top layer, these cells are pretty flattened. And they're all alive. You can still see nuclei all up and through here. So these cells are alive. They're flattened. So this would be stratified squamous. Squamous, squashed nuclei. See our next slide. Now here the pointer is in the lumen. Remember, epithelial tissue always faces the lumen. So here would be our epithelial tissue. The basement membrane is here, and this is the connective tissue, and through here. So this is our epithelial tissue. Even on this mag, you can pretty much tell these are columnar cells. Look at the nuclei here. They're elongated. Remember, columnar cells, their nuclei are elongated. They look tall, just like the columnar cells themselves. Let's go a little bit higher. So this is going to be simple columnar, one layer thick, columnar cells. Here you see some goblet cells spread out through here. And here you see kind of a fuzzy border on the apical surface. Oops, oh, I don't know how to get there. This fuzzy border, this is microvilli. Microvilli are cytoplasmic extensions that increase the surface area of the cell. And that's for absorption. So this is probably going to be from the small intestines here. So simple columnar with microvilli here and goblet cells. Remember, goblet cells make mucus, and in the digestive system, in the GI tract, the mucus is there to lube the tube. Okay, on this slide, you've seen this in class. You should just by looking at this, even if you don't know what this structure is, we'll get to know it later when we get to the urinary system, but this is from the kidney. You look at this and you go, oh yeah, I remember seeing this. There's this big glob with this white thing around it. Well, this white thing is the lumen. So let's look closer because epithelial tissue is always facing the lumen. So the pointer is in the lumen. Here's the epithelial tissue. This is one layer thick. One layer thick. Squashed nuclei. See it going all the way around here? This is simple squamous. Simple squamous. One layer thick, squashed nuclei. There's going to be lots of simple cuboidal in this slide too, but for now we're just concentrating on this simple squamous right here. So pointer again in the lumen. The cells, epithelial cells, always face the lumen. Let's get a little bit higher. So again, here we see the apical surface. The basement membrane is going to be following around here. And all this stuff in between is the connective tissue. So even from here, you can pretty much tell this is simple columnar. Simple columnar. Let's get a little bit closer. Simple columnar. There's some goblet cells in here too. But this is simple columnar, and you can kind of see this fuzzy border on the apical surface. This is microvilli. Some people call it a brush border. Remember, microvilli increase the surface area for absorption. Now the pointer's not quite in the lumen here. Here's the lumen, and here's going to be our epithelial tissue right here. So the basement membrane is going to be around, around like this. Around like this. So it's more than one layer thick. So let's look at those apical cells and see what they look like. Here's our lumen. Look at these apical cells. These are weird looking cells. These dome-like cells. This is going to be transitional epithelium. Found in the bladder and the ureters. They carry urine, so when the urine fills up this lumen, it's going to compress on these apical cells and squish them, so they are going to look flattened. So when the bladder's full, these apical dome-shaped cells flatten out. That's why they call it transitional. These cells on the apical layer transition from these dome-like cells to flattened cells when the bladder's full. Oh, looks like we've got another transitional. Let's see. So here's the basement membrane somewhere down here. And we have all this layer, so we know it's stratified. You look at the apical surface, here's the lumen. These weird cells again are up in here. So once again, this is transitional. Transitional epithelium. The pointer is in the lumen. Epithelial tissue always faces the lumen. So here's going to be our basement membrane. Pretty easy to see here. Apical surface here. This is all connective tissue. So how many layers thick? Well, this one, here you see the nuclei all over the place. You'd go, wow, it looks like it's more than one layer thick. But it's not. This is pseudo meaning false. Pseudostratified, columnar. These are columnar cells. Look at the nuclei. They are elongated. Elongated nuclei, columnar. It's got cilia. See the cilia here? Cilia much different looking than microvilli. Cilia are hair-like extensions that move. They are motile. They move whatever on top of their surface. So this is going to be pseudostratified, ciliated, columnar epithelium. And also, this is the connective tissue below, and we said it's the connective tissue that brings oxygen and nutrients to our avascular epithelium. Well, here's a blood vessel. Here's a blood vessel. Actually, here's another blood vessel down here. You can see some red, that's red blood cells in the blood vessel. Here is flattened nuclei here going around the blood vessel. You can see flattened nuclei here too. So this is simple squamous. Remember the type of simple squamous that lines the blood vessels? What was that called? That is endothelium. Endothelium, simple squamous that lines our blood vessels. Now this slide is something they call a hole mount. They are taking the epithelial tissue and they are placing it on top of the microscope slide, so you are actually looking down on the tissue. You're seeing this whole sheet of epithelial tissue. So this is the looking down on the tissue. So I showed you this in class. Here are the individual cells. You can see there's no space in between these cells. Cellular, very classic for epithelial tissue. This is mesothelium. Mesothelium. Remember what mesothelium is? Mesothelium, a special type of simple squamous that this forms our serous membranes. Remember our serous membranes? So mesothelium, a special type of simple squamous that forms that. This is a special type of simple squamous, and it's found on serous membranes. Mesothelium. Again, the pointer is in the lumen. You can pretty much see the cells around there. Let's go a little bit higher. Lumen, here's the apical surface. So we've got a tube here, a round tube. Here's the basement membrane here. So, nice round nuclei. One layer thick. This is simple cuboidal. You see a lot of them. Simple cuboidal. Simple cuboidal. Simple cuboidal. Simple cuboidal. Lots of simple cuboidal in this slide. Pointer in the lumen. Multiple layers thick, so it's stratified. Look at the top layer. these cells are squashed. You still see the nuclei in these cells up here. So this is living. These cells are all alive. So this is stratified squamous. Oh, I hate when I do that. Oh, well, I can't. There, stratified squamous. Now, when we get to the integumentary system, we'll learn the difference between keratinized and non-keratinized stratified squamous. This is non-keratinized stratified squamous. The cells on the top layer are still alive. This is going to be found in areas of wear and tear. You need lots of protection. You're going to find this in your esophagus. You're going to find this in the back of your throat where food goes down. You're going to find this in the anus. And you'll find stratified squamous non-keratinized in the vagina too. So any area that has this exposed to wear and tear. Stratified squamous non-keratinized. Pointer in the lumen again. You can already tell, or at least you can see that the apical region up here, it looks like cilia to me up here. Let's go closer. Yep, look at that cilia. Real easy to see. Does not look like microvilli. You can see the individual hairs up here, the cilia. This is going to be pseudostratified ciliated columnar epithelium. Here's a little goblet cell right here. Now this is skin. This is, when we get to the integumentary, you'll be looking at this. Here is the epithelial layer. Starting down here, the basal layer, it goes up and down. Here's all the basal layer. This is all connective tissue, the light pink stuff below. So the basal layer is here, and it goes from here all the way to the top. This is the apical surface that is facing the lumen. Here's the lumen. Let's go on high. Lots of cells, lots of cells. And you can see it gets really dark up here. This is where the cells of the skin, they actually start dying up in this layer here. So when we get to the integumentary, we'll learn about these different layers. But up here, do you notice anything different about these cells? They have no nuclei. No nuclei. They are just empty cells. These cells are all dead. Dead. Dead as a doornail. This is stratified squamous keratinized. So once we say it's keratinized, these cells have filled up with a protein called keratin. We'll talk about that when we get to the integumentary system, and they have since died. But this is stratified squamous keratinized. Now, when you see this slide, immediately nothing else looks like this. You know the organ this is from. This is from the lung. Nothing else looks like that. So the pointers in the lumen, each of these little pockets is an alveoli. Alveoli is where gas exchange takes place in the lung. It's going to be simple squamous. You want diffusion of gases. You want it as thin as possible so that those gases can diffuse quickly through that thin, simple squamous membrane. Let's see, let's go a little bit closer. Simple squamous all through here. Simple squamous. Remember, lung, nothing else looks like this. Holy gamoly! What do you see here? Here, the pointer's in the lumen. Here's the epithelium. So it's more than one layer thick, so we already know it's stratified. Let's look at those top cells. Holy gamoly! These are the weirdest looking cells. Aren't these just the weirdest looking things? What kind of epithelium is this? This is transitional. Bladder and ureter. Here's the lumen. This is where the urine's going to be, in through here. Here's another picture of the kidney. So we're looking at the lumen again. Go a little bit closer. Again, pointer in the lumen. Here you can see this ring of simple squamous. Simple squamous. Hopefully you can see it. There's the nuclei. Flattened nuclei, one layer thick. One layer thick. Here's the lumen, simple squamous. And our last one. Oh, an easy weasy one. Pointer in the lumen. Here's the basement membrane. Nice round nuclei. This is simple cuboidal. So I hope this helps you going through it. They're not that bad. You just have to get used to seeing them and you'll figure them out pretty quickly. Hopefully this helps, and if you have any great pictures that you took yourself on the through the microscope, please send them my way. Thanks.